Thank you, Martin. Yes, so I'm Joel Tabb, and I'm with IONICA Sciences, all the way from Ithaca, New York. And we are developing a rapid, sensitive system for detecting Lyme disease. So IONICA Sciences, we combine several new technologies to create a new diagnostic system. We're combining DNA aptamers, which will replace antibodies for binding, as well as a technology known as surface-enhanced Raman spectroscopy to create this sensitive, highly specific platform for infectious diseases. And our first test is for Lyme disease, which I think most everybody here who lives in this part of New York knows all about, probably knows somebody who's had it or had it themselves or worried about having it. Over 300,000 people are diagnosed annually with Lyme disease, and it causes about over $3 billion in costs related to lost productivity, healthcare costs. And one of the major problems is that the current tests are really awful. They have about a 40 to 60% false negative rate. They have poor sensitivity, meaning that if you come back with a negative result, there's over almost a 50% chance that you still have the disease, meaning that there's a lot of uncertainty and a lot of undiagnosed Lyme disease going on in the country. So as I said, these tests have poor sensitivity, high false positive rates, and another big issue is they monitor for the development of antibodies in your body, so they actually detect the body's response to infection, not the actual disease. So once you have been exposed, your antibody levels are high, you can't retest. Okay, so once you've been exposed, that's it. You've got a result. You can't determine efficacy of treatment. You can't follow up for reinfections. And really, a high sensitivity direct test has the potential to really dramatically change the landscape for Lyme disease testing. We think increasing the market fivefold or more. So what currently goes on? Well, you're out doing your thing. This guy likes to lie out in the grass, hang out. He's looking at the sunny days. This is clearly not Ithaca. But so what happens? You start to feel bad. You wonder, you're starting to feel joint pain or something. You go to the doctor. Well, the doctor starts to ask about travel history, clinical history, where you've been. Maybe you're not from this area, but you traveled up here. And they decide to maybe run a Lyme test. Well, it's a two-stage test they run. The first is an immunoassay, fairly standard technology. This determines if you've likely been infected, and then a laborious Western blot. What happens is, well, there's still this 50% chance that you don't know if you've got the disease or not. Your sensitivities come back. And so essentially, it's so unreliable, you don't know what to do, and many physicians don't even bother prescribing the test. They rely on either their own clinical expertise, or if you're in an area that doesn't know about it, you actually go undiagnosed. So this is a major problem. So what have we done? We've created a simple test for detecting Lyme disease. Essentially, you take a finger prick sample, you put it on our special sauce system. These are DNA aptamers connected to our proprietary surfaces. And this allows for a rapid response that we can then monitor with an optical signal. We can monitor and analyze the spectrum. So you can go from sample collection, a finger prick, a serum sample, put it on a self-contained cartridge, stick it in your analyzer, and within 20 minutes the doctor has a result. Therefore, it really dramatically changes. I want to be clear, though, what we've developed here a platform, an ability to detect infectious diseases, and the first test, the proof of concept, is for Lyme disease. We've submitted a patent for this application, and this is our own patent. We are not associated with it. Although we're at a Cornell incubator, this is our own technology. So how does the system work? It's a two-component system. There's a disposable cartridge where you put the blood sample in, you do the detection work, and you get rapid results, either positive or negative. As I said, you can use whole blood. Binding takes under 15 minutes, and it's simple to use. Take the sample, put it on the cartridge, insert it, get results. There's also a device, an actual instrument that's involved. It's a system that's designed as a clinical application of a Raman spectrometer. And this, most importantly, this allows for very high sensitivity detection. By using Raman spectroscopy, especially surface enhanced, we have between 1 million and 10 to the 6th and 10 to the 12th fold amplification of the signal. So we have a very high sensitivity test here. So that eliminates false negatives. It's high specificity, so you low false positive rates, and essentially there's no training required. So this is great for a clinical laboratory because it will not dramatically change workflow in that environment. So how does this work? Well, you think you're lying out in the grass. You think you don't feel well. Well, they run a test, they use our instrument, our chips, and if it's negative, you go about your business. You don't worry about it. But if it's positive, they can prescribe a not over the counter, but a generic antibiotic prescription, and you've been treated now for Lyme disease. The advantage of this test is it theoretically can be done more quickly than both faster, but also there's no lag time for antibody development in the human body. You can run a test. Current tests require 6 weeks post-infection. This should be able to be done much faster. It can differentiate between old and new exposures. So many people have been exposed. They look positive, but they actually don't have the infection now. Also, people get reinfected. If you live in an area and you do hiking, you're out and about, you visit the Adirondacks, you will be exposed multiple times. And it also will prevent the need for prophylactic antibiotics when you don't know if you've actually got Lyme disease. Our test, as I said, it has the potential to be portable. We're actually proposing to use this in a clinical laboratory environment for the moment, but it can be portable. It's fast. It has high sensitivity. No sample prep, so that's ideal. You're actually just taking a drop of serum or blood, putting it on. No training. It's great for a clinical laboratory. No maintenance. And one of the major advantages, it's a single-stage test. So there's one test as opposed to requiring 2 tests. So what's the market like? Well, the current market is the current test that is in the market is actually an LDT, a laboratory-developed test, meaning they're not FDA-cleared tests, and they're these 2-stage tests. Therefore, no one company dominates the marketplace. They're done at clinical laboratories around the country. 3.4 million tests are ordered annually in the U.S. for a market of about $300 million. But we think by changing the way the tests are done and actually having a specific rapid test that directly detects Lyme disease, we can dramatically increase this market to over $1 billion. And this is based upon the fact that as clinicians aren't prescribing the test, and a direct test could allow for screening, for retesting, and to measure the efficacy of treatment. So our business model is, as we said, there's 2 parts to this model. There's the instrument and the disposable Lyme cartridges, the detection cartridges. Our product will be sold as what's known as a reagent rental paradigm, where you're putting the instrument. The instrument will be placed in clinical laboratories free of charge in return for a contract for specified numbers of tests to be run. This allows them, therefore, we're responsible for replacing instruments, we're responsible for upkeep, and they're not buying an instrument that they may not be using down the line. The other advantage is this will be an FDA-cleared test. So we're prepared to go to the FDA and sell this through distributors instead of using a laboratory-developed model. This is a major advantage. It'll be the first FDA-cleared Lyme test on the market. And an additional advantage is this is the first test of many to be developed for this platform. So this platform can be used for a wide variety. Once this is demonstrated for Lyme disease, to use for a wide range of infectious diseases. This is a lot to go through, but the idea is we put a plan together to get this through the FDA, and this involves where we've actually just closed this past week a seed stage round for funds from the Global Lyme Alliance, an advocacy group for Lyme disease, to fund preclinical studies. We've put together already a manufacturing plan to develop prototypes and devices, but because there's a long lab time through the FDA, we're proposing to also develop a veterinary test. Same technology, same test could be used and sold with a much more rapid route to development and faster to sales, and allowing for actual tests on the market. But in addition, in a parallel route, we would go to the FDA and develop a plan for pre-sub and FDA trials and clearance. The goal would be to have veterinary tests on the market in 2 years and FDA cleared tests by the end of 2020. So our team is co-founders are Omar Green and I. Omar's a chemist and I'm a biologist. We have extensive experience in the biotechnology world. Most of our business advisors come out of through the McGovern Center Incubator at Cornell University. Bill Rhodes is an executive in residence there. He's out of Bennett Dickinson. He was the president for BD Biosciences, has extensive experience in the diagnostic world. Becky Robertson is a VC out of California. She's funded a lot of in vitro diagnostics company. And Lou Walser is the director of the McGovern Center. They're all business advisors for us. We also have technical leads. Patricia Delamore is a Lyme disease expert out of New York City, and John Lombardi is our spectroscopy expert. So we think we're primed to really change the Lyme disease market. This test, current tests are inadequate, and we think that it's clear that poor Lyme disease tests lead to really serious problems. Arthritis, carditis are infectious of the heart, encephalitis, neuropathies. The undiagnosed Lyme disease is a real problem. Our test, what we call the IO-Lyme test, was expected to be superior, especially in terms of sensitivity, time, and ease of use. And we're working with an advocacy group to get doctors and patients to understand our technology, as well as working with insurance companies to put together a reimbursement plan. So as I mentioned, this test can be used for a variety of pathogens. In addition to Lyme disease, we're looking at the next step would be to develop other tick-borne disease tests to create a panel. Mosquito-borne diseases are exceptionally important. You've heard of Zika. Dengue is one of the major infections, infectious mosquito-borne infections. And surprisingly, a big problem is that I wasn't aware of are STIs, sexually transmitted infections. Turns out syphilis is a related bacteria that causes Lyme disease. All of these can be developed on the same platform. So with that, this is our test, and I guess we don't have questions. So there we go.